Good evening. In the module on biotechnology, I have uploaded another file on polymerase chain reaction. It is a technique that was developed by Gary Mullis in 1983 for which he was awarded Nobel Prize. Polymerase chain reaction is a technique that is used to make copies of a DNA fragment. You can call it as a chain reaction because one DNA fragment produces two produce four, and so on. Generally, PCR is run for 25 to 30 cycles at one go, and after about 25 cycles of DNA synthesis, about 10 to the power 5 copies of the specific target DNA sequences are produced. These DNA fragments are then run over gel electrophoresis, where they are visible as a discrete band of a specific size. The piece for running a PCR cycle, we require a template DNA, two primers which are known as forward primer and the reverse primer, a thermostable DNA polymerase, that is Taq polymerase, and DNTPs. There are three major steps that are followed in PCR. The first step is denaturation of the DNA template. For denaturing the DNA, the mixture is heated to 94 degrees Celsius so that the hydrogen bonds that hold the two DNA strands together, they are broken and the DNA denatures and we get single-stranded DNA fragments. Second step is annealing of the primers. For annealing, we have to lower the temperature to 50 to 60 degrees Celsius. The complementary DNA fragments, template DNA fragments, they can also anneal to each other, but the primers which are having the complementarity with the region of the DNA template can also bind. So we get hybrids of two kinds. One is the hybrid between the original DNA template strands and the hybrid that is formed by the DNA template strands and the primers. So if there are more number of DNA primers, so there are more chances that the hybridized DNA fragment and the primer molecules, they are formed. The third step is the extension of the new DNA strand. For extension, we need the action of Taq DNA polymerase, which works very efficiently at temperature of 72 to 75 degrees Celsius. So we have to raise the temperature. So after that, DNA polymerase binds to the primers and then extends or forms the new DNA strand, thereby giving two double-stranded DNA molecules from one single-stranded DNA